Hello, Todd Dunn here for Life on the Edge. This presentation is CLL Series number 5, for CLL is chronic lymphocytic leukemia. The topic of this presentation is, what is a FISH test? Anybody who has CLL or knows someone with CLL has probably heard about this test, and what I hope to do today is explain a bit more about what the test can tell you, how it's done, what its limitations are, and finally at the very end just how much this test costs. FISH in FISH test is an acronym standing for Fluorescent In Situ Hybridization, or FISH. FISH testing identifies chromosomal abnormalities, in the case of CLL, in B lymphocytes or B cells. The test looks for specific abnormalities using chemicals called FISH probes, and one of the limitations of the test is it only identifies the specific abnormalities that are tested for. In other words, there might be other abnormalities there, but a FISH test won't detect them if they aren't on the list of things that the test is specifically looking for. The way a FISH test works is by putting chemicals called FISH probes into a sample of blood. What is a FISH probe? Well, a FISH probe consists of two parts. Part one is a small piece of DNA which is complementary chemically to a particular location on a chromosome. That piece of DNA is then attached to a secondary molecule which is designed to emit a particular color of light when exposed to fluorescent light. This combination of this little piece of DNA that matches a spot on a chromosome and the fluorescent emitter is called a FISH probe. To make my explanation of a FISH probe a little bit clearer, let's consider the diagram shown here. On the left is my stylized strand of DNA. In the center is a FISH probe. The little green part matches up with a particular spot on the DNA strand. The red part of the FISH probe is the fluorescent light emitter. When the FISH probe is mixed in with the DNA from the sample, it hybridizes or binds to the DNA at the particular spot that it matches, as shown in the right-hand side of the diagram. The FISH probes used for CLL testing are called a CLL FISH panel. A FISH panel consists of a number of different probes to look for specific chromosomal modifications, or in the case of CLL, primarily deletions of segments of the chromosome. The list here shows the particular probes. They are typically a 6q probe that looks at the long arm of the 6th chromosome, typically at locations 6q21 and 6q23. The 11q probe, which typically is going to look at, again, the long arm of the 11th chromosome at position 11q22.3, and this is also called an ATM probe. Then there's a probe that looks for the presence of the 12th chromosome, and it just binds to the 12th chromosome. Then there's the 13q probe, which there are often two different 13q probes used in a FISH test. One binds at 13q 14.3 and the other one at 13q 34. And finally, there's the 17p probe, which binds at 17p 13.1, which is also the location of the p53 gene on the 17th chromosome. These numbers, 11q22.3 for example, are specific locations on the chromosome where the probe will bind. The way the test works is these probes bind to the chromosomes if the chromosome is complete. If the chromosome is not complete, then obviously the probe cannot bind to the chromosome. So it won't produce a signal, which I'll be talking about in a minute. In addition to the probes listed here, there's usually also a reference probe, which is just used to determine whether or not the FISH probes are working properly and are hybridizing or binding to the chromosomes of interest. So how is a FISH test done? What is the procedure? Well, it's actually a relatively straightforward test, although some of the specific steps are a little bit complicated. I'm not going to go into the details, of course, but I will outline the major steps. Step 1. A sample of B lymphocytes or B cells is prepared. It's necessary to separate out the B cells from the other lymphocytes because under a microscope you really can't tell the difference between B and T cells, so you have to be able to differentiate them or you'll get inaccurate results. So B cells are separated from the other lymphocytes and prepared as a sample. The second step is the B cells are fixed to a glass slide so that they will stay on the slide and not move. At that point, the probe molecules are added to the sample. Now I should point out that sometimes several probes are added to a particular sample, but usually there's more than one sample because not all the probes emit different colors of light, so you don't want two probes that both emit, say, red light in one sample, or you won't be able to tell which one you're looking at. So B cells are attached to a glass slide, probes are added. The next step is to do what's called denaturing the sample, and basically this is simply taking the sample and briefly heating it to a relatively high temperature, which causes the DNA strands within the sample to separate from the chromosomes and from each other and become available for the probe molecules to react with. This denaturing step usually uses a temperature of about 75 degrees Celsius or about 140 degrees Fahrenheit, 150 maybe, and it's very short, only a few minutes. Once that's done, the sample is incubated and for usually overnight, 12 to 18 hours, at 37 degrees Celsius, which is essentially normal body temperature. And this is to allow the probe molecules to hybridize to the DNA strands, to attach to them. And so this is carried on for about 12 to 18 hours. After that, the sample is washed to remove excess probe molecules, because if they're present in the sample but not attached to the DNA, they'll still give a signal when we expose them to fluorescent light, so we want to get rid of them and only have probe molecules in the sample that are attached to DNA strands. The final step is to take the sample, put it under a fluorescent microscope where the individual B cells can be seen, and then the presence or absence of probe molecules shows up as points of light, and they can then be counted and determined whether or not each cell has the correct number of probe signals for unaltered DNA, or if it doesn't, it suggests that there is a DNA deletion, or in the case of trisomy 12, there might be 3 signals instead of the normal 2 that you would expect, since each cell has 2 copies of each chromosome. So that's the procedure, pretty simple, and at least in theory. In practice, it's quite a bit more involved. In order to make the fish sample analysis a little more comprehensible, I'm going to talk about these diagrams here. So when we look down at a sample through a fluorescent microscope, which is shining fluorescent light on the sample, the probe molecules that have attached to the DNA in the sample will emit their characteristic color of light. For the example I'm using here, it's red. If both copies of the chromosome in the particular cell have the segment of DNA present that the probe attaches to, you'll see two signals, as in the left cell I've shown here, two red dots. If, on the other hand, one copy of the chromosome of one chromosome has a segment deleted, the probe cannot attach to that chromosome, so only one chromosome will have a probe attached to it, and we'll see only one signal. This is called a heterozygous deletion. And finally, if both copies of the chromosome have the segment of the chromosome that the probe attaches to missing, there will be no signal, and that's called a homozygous deletion. For FISH analysis of chromosome deletions, the technician looks through the microscope at each cell and determines if there are 2 spots for each probe, 1 spot, or no spots. If there are 2 spots, the deletion that they're looking for, let's say it's 13q, is absent. If there's 1 spot or no spots, the deletion is present and would be noted as, the cell would be noted as 13q deleted, because at least one sample of the chromosome or both samples of the chromosome are missing the spot where the probe would have attached. In a more sophisticated analysis, they might also make note of whether or not it's a heterozygous or homozygous deletion. Typically, in an analysis, at least 200 cells are counted, which gives pretty good precision for the analysis. If, for example, 50 cells show up with either a homozygous or heterozygous deletion, then we would say out of 200 cells that are counted, then they would report the sample as having 25% of the cells showing the particular deletion that the probe identified. Okay, now that your FISH test is done, how are the results presented? Typically, as shown in this slide, what you'll get is a list of the different things that the FISH test was looking for. In this case, deletion 6q, deletion 11q, plus 12, which is trisomy 12, deletion 13q, and deletion 17p. This happens to be my personal FISH test result. In my test, there were no deletions of chromosome 6q detected. In other words, every cell they looked at had 2 signals for the probe for chromosome 6. Same thing for deletion 11q. Every cell had 2 signals for deletion 11q probe. But for deletion 13q, 17% of the cells that they looked at had either one copy of the chromosome segment where the probe attached missing, or both copies. The results did not differentiate between heterozygous and homozygous deletions. So if they counted 200 cells, in my case, 34 of them only had 1 or no signals for the 13q probe. And finally, for 17p deletion, all of the cells they looked at had 2 signals indicating that there were no deleted segments of 17p. In addition to these results, your FISH results sheet will also have a list of the probes used. This list usually gives the specific names for the probes that were used. The names are specific to whichever company provided them. and there also will be a brief methodology summary, which in the case of my results simply said they counted 200 plus cells, and that was pretty much it. So that's it. That's what your FISH results look like. It's pretty simple. Usually after you get the test, the results will be passed on to a pathologist who will write a paragraph summarizing the results and the implications of the results. And that's what you will get back. Now that I've finished going over the procedure for a FISH test, I want to say one other thing about the test. That is a fairly important thing to remember. The FISH test can only detect what it's looking for. So if you had a deletion in a chromosome that no FISH probe was used for, the FISH test would not detect it. So the FISH test is only looking for certain things, and that's all it can detect. It's a very specific test in that respect. So, and it is a very sensitive test, but it is also limited to what the specific design of the test was. Okay, now let's get on to the really important part about a FISH test. That is, how much do these things cost? We know that having CLL can be very expensive, and a FISH test, which pretty much everyone has at one stage or another, and sometimes more than once, is one of the cost drivers. So I was unable to find anything online about FISH tests, so what I'm going to present here is how much my FISH test cost. Now mine was done through my local hospital. They sent the samples out to a laboratory that did the test. So here are the costs. First, I'm going to give you what the hospital actually billed me. They billed me to draw the blood for the FISH test. They drew about 8 vials of blood that day, and not all of them were used for the FISH test. I don't really know how many of the 8 were sent off for the FISH test, but they charged $31 to draw my blood. So some of that $31 went toward the FISH test. The FISH test itself was billed at $3,264. And finally, the pathology report that came along with the FISH test was charged at $288 for a grand total of $3,583. So if you happen to be unfortunate enough not to have insurance and you went to my hospital and got this done, you would be expected to pay $3,583 for this FISH test. If you had more than one during the time that you had during your CLL treatment period, you would be paying this over and over again. Fortunately, I had insurance, so my insurance only allowed part of the costs. For the blood draw, they allowed $28.05. The test itself, they allowed $2,488.04. And for the pathology, $244.80. So what the insurance allowed of the billing was $2,760.89. So still quite a lot of money. Now, if you had Medicare, this was commercial insurance. If you had Medicare, these numbers would be smaller because the cost, Medicare doesn't pay quite as much as commercial insurance does. Also, you wouldn't have to worry about it because diagnostic testing like this is covered at 100% by Medicare, so it isn't an issue if you have Medicare. At any rate, as you can see, the FISH test is pretty expensive, at least where I got it done. You might expect it to be perhaps a little more in some places or a bit less in other places, just depending on the particular lab that did the test. Okay, that finishes up what I wanted to say about the FISH test. I hope that what I've said today clarifies the test to people. They now know a little bit more about what's involved, what the FISH test can and cannot tell you, how it's done, and how the results are generated. So that is all I want to say. I hope that this has been useful to you. If it has, click the like button or subscribe if you want to get notifications of what's coming next. Thank you very much.